Krebs cycle is the second part of cell respiration, which is the process that cells use to break down sugar for ATP. Now, in our first part, glycolysis, glucose was broken down into two molecules of pyruvate, and we know that pyruvate is made up of 3 carbon, but pyruvate cannot enter the Krebs cycle. Instead, it has to be converted into acetyl-CoA. And the CoA is just a coenzyme A that's going to help bring this acetyl, which is then a 2-carbon molecule, to the matrix of the mitochondria. Now, as it's converting pyruvate to acetyl-CoA, it's going to release the gas carbon dioxide, and that's going to be 1 per each pyruvate that's going to be converted. Krebs cycle is aerobic, which means oxygen needs to be present for this to take place. If oxygen is not present, pyruvate is going to go through fermentation. So instead of being converted into acetyl-CoA, it's going to be converted into lactic acid or ethyl alcohol. So if we're looking at our Krebs cycle, our very first step is this acetyl-CoA is going to be brought into the matrix of the mitochondria. Now, this coenzyme A is then going to leave and then help break down another pyruvate and bring it back to the matrix. So CoA is then able to leave. This acetyl-CoA is going to bind to a 4-carbon molecule called oxoalloacetate. And I don't have an acronym for this, so we're just going to have to know this big, long word. So oxoalloacetate, I guess we'll keep adding our numbers here. So acetyl-CoA comes in, CoA is free to leave, and the acetyl is going to bind to this 4-carbon oxoalloacetate. So when it comes in and binds, we know acetyl has 2 carbon, plus our 4 over here. This is going to make a 6-carbon molecule called citrate. So number 4, this makes citrate. Now this citrate is going to be modified, and we see our electron carrier, NAD, is going to come in, grab those electrons, and it's going to turn into NADH. NADH is then free to take these electrons to the electron transport chain, where they're going to be used to further complete cell respiration and make energy. When these electrons are picked up by NAD and it turns into NADH, CO2 is going to be released. And we see that this happens again. When this CO2 is released, starting from a 6-carbon, we're down to a 5-carbon molecule. We're going to reduce it again. NAD is going to come in, take electrons, turn into NADH. It's a plus there. And again, go to the ETC. When these electrons are taken, we're going to release more CO2. And when this CO2 leaves from a 5-carbon, we're back down to a 4-carbon molecule. This 4-carbon molecule, we're going to be able to store some energy from it. So we see ADP, our low energy molecule, is going to store some energy, and it's going to turn into ATP. Now this 4-carbon molecule we're going to regenerate into oxoalloacetate. We see that FAD is going to come in. FAD is another electron carrier. It's going to pick up electrons in a hydrogen, and it's going to turn into FADH2. FADH2 is going to take these electrons to the ETC. And then we have more electrons being taken. Keep up with my number here. That's 13, step 14, step 15. We have NAD plus comes in, takes electrons, and it turns into NADH. And again, these electrons are free then to go to the ETC. So if we just walk through this one more time, Krebs cycle is aerobic, so oxygen has to be present. and it's going to occur in the matrix of the mitochondria. Now, if oxygen is not present, pyruvate, made at the end of glycolysis, is going to be converted into lactic acid or ethyl alcohol through fermentation. But if oxygen is present, pyruvate is going to be converted into acetyl-CoA, going from a 3-carbon molecule to a 2-carbon molecule, and we're losing one of those carbon in the form of carbon dioxide. So our first step of Krebs cycle, acetyl-CoA comes in, and the CoA is free to leave. This acetyl, which is a 2-carbon molecule, binds to the 4-carbon molecule oxaloacetate, and that forms citrate, which is a 6-carbon molecule. NAD plus is going to come in and take electrons, grabs the hydrogen, turns into NADH, goes to the ETC, carbon dioxide is released. We see this step happens again. NAD plus takes electrons, turns into NADH, goes to the ETC, carbon dioxide is released. So going from 6-carbon, releasing a carbon dioxide, down to a 5-carbon molecule. Same thing, release a carbon dioxide, we're down to a 4-carbon molecule. Energy can be stored. We just make 1 ATP. And then we have another electron carrier, FAD, takes electrons, turns into FADH2, goes to the ETC. And then take a little bit more electrons, NAD plus to NADH, grabs electrons, goes to the ETC, and this regenerates our oxaloacetate, which is a 4-carbon molecule. Because glycolysis makes 2 pyruvates at the end, and these 2 pyruvates are converted into 2 acetyl-CoA, the Krebs cycle is going to take place twice for every glucose. So 2 times for each acetyl-CoA that is going to be generated. And that's our Krebs cycle.